I will start with the title, which is Who is Linus Pauling? So first of all, I will tell about his background history. So basically, number one, who is he? So basically, Linus Pauling is an American chemist, biochemist, chemical engineer, peace activist, author, and also educator. Next one, where is he from? So basically, he was born on February 28, 1901 in Portland, Oregon. So the next one is, what does he do? Which I mean is, what is his occupation? So he was one of the founders of the field of quantum chemistry and also molecular biology. He published more than 1,200 research papers and also books, which is about 850 dealt with scientific topics. Next one, his academic background. Basically, he graduated from Oregon Agricultural College. Now, it is well known as Oregon State University in 1922 with a degree in chemical engineering. He then received his PhD in chemistry from the California Institute of Technology, Caltech, in 1925. Last but not least, why did he contribute as geneticist? He contributed as geneticist by studying the molecular basis of human diseases, such as sickle cell, anemia, and proposing the concept of molecular disease. He also developed the theory of electronegativity and the scale of electronegativity of the element, which is a useful tool for predicting the nature of chemical bond and the structure of molecular one. Last one, what made him interested in genetics? He became interested in genetics after reading the book of The Mechanism of Mendelian Heredity by Thomas Hunt Morgan in his college in 1926. He was fascinated by the idea that the genes are responsible for the inheritance of traits and that they are composed of molecules that can be studied by physical and chemical methods.
Continuing with the third point, how did he discover it? The first documented case of sickle cell anemia was in 1910 by a physician named James Herrick. A 20-year-old college student was described as severely anemic. The blood smear of the patient showed that the shape of the red blood cells was very irregular and the large number was thin, elongated, sickle-shaped and crescent-shaped forms. This then caught the attention of Linus Pauling in 1949. Since the hemoglobin is abundant in red blood cells, he thought it would be worthwhile to experiment and examine the properties of hemoglobin obtained from sickle cells. This led to the discovery that both the oxygenated and deoxygenated forms of sickle cell hemoglobin had higher isoelectric points than those from normal erythrocytes, suggesting that sickle cell hemoglobin is more positively charged than normal hemoglobin.
Okay, for the next point, does he conduct any experiment regarding this? So, Pauling used his knowledge about the X-ray crystallography and his understanding on chemistry rules to determine how amino acids fit together to make a protein. After using a paper to make a model working with jigsaw puzzle, he eventually figured out the structure of alpha helix of globular protein. Pauling later performed a technique comparing protein among people with sickle cell, without sickle cell, and carrying a gene for a sickle cell. He found that the hemoglobin of those with sickle cell differ from those without it. The carrier of sickle cell had both types of hemoglobin.
The methodology that he used is molecular model building, which visualized and understood interaction between molecules. Quantum chemistry that explained the formation of the hydrogen bonds and other key interactions. The collaboration is to share ideas, gather data, and refine research with other scientists. Critical thinking and analysis is being questioned by existing theories and propose new explanations. And also, communication effectively shared discoveries through publications and lectures.
We go to the objective of his experiment. 1. Understand the nature of the chemical bond and its role in molecules. 2. Unravel the structure of protein and their function. Number 3. Find the cause of sickle cell anemia and explore potential treatments. 4. Discover the structure of DNA and unlock its genetic secrets. Number 5. Unleash the power of science to improve human life.
Yes, he did succeed in discovering sickle cell anemia in November 1949, but he failed to predict the structure of the DNA. He proposed a triple helix structure with bases on the outside. The problem with his triple helix structure is that the phosphate from the helical core, with the base pointing outwards. This will be impossible under normal cellular condition. Each phosphate group is negatively charged, and so many negative charged forces forced together would repel each other, literally driving the structure apart. So, what challenge did he face? The model Pauling put together did not seem to match a critical measurement, Asbury 5.1 picometer repeat, which would theoretically measure the distance between each full turn of the helix. Pauling knew that much more careful model building would have to take place before it was confirmed one way or another. But for the moment he held off spreading the news, he did not write query about it. No records indicates that he mentioned it to any of his British hosts. He did not even save his original sketch. Instead, he filed the idea away. So, the second challenge was in 1964, Linus Pauling left his tenure professorship at Caltech because of pressure from administrator and conservative trustees who disapproved of his prominent, persistent anti-nuclear and international peace-promoting activities.
How did he introduce it to the world? Number 1. Scientific publication. Pauling wrote over 1,200 scientific papers that explain his research for other scientists to use and wrote on. 2. Through books. Pauling wrote several books, including The Nature of the Chemical Bond, which explain his ideas about chemical bonding and how they relate to biomolecules. These books are very valuable resources for students and scientists. 3. Lectures and conference. Pauling was a gifted speaker and often presented his research findings at conferences and universities worldwide. These lectures allow him to share his ideas with a broader audience and engage in discussion with other scientists.
This lecture is the first of 3 lectures on one part of chemistry, valence and the structure of atoms, molecules, and crystals. It was the British scientist Eddington who said that the study of the physical world, and I would say the biological world too, is a search for structure and not a search for substance. If we want to understand the human body, we must know its structure in terms of the cells that make it up. If we want to understand cells, we must know their structure in terms of molecules. If we want to understand molecules, we must know their structure in terms of atoms. And to understand atoms, we must know their structure in terms of electrons and nuclei. One nucleus down in the center of the atom with a number of electrons around. To understand nuclei, as the physicists are trying to do now, we must learn their structure in terms of protons and neutrons, and perhaps mesons too. And it may well be that at some time in the future, the fundamental particles, the electrons, protons, neutrons, mesons, will be found to have structure also in terms of still smaller particles that have not yet been discovered.
Number 4. Media interviews and outreach. Pauling actively participate in media interview and public engagement activities, explaining his work in a way that was accessible to the general public. This helped raise public awareness about scientific discoveries and inspire future generations of scientists.
Dr. Pauling feels that his experiments in X-ray crystallography helped develop his understanding of the atom.
I was very fortunate in having A.A. Noyes suggest to me or tell me that I was to work with Roscoe Dickinson on X-ray crystallography, determination of the structure of crystals by X-ray diffraction. This technique gave for the first time detailed information about how atoms are related to other atoms in a crystal and how far apart they are from the other atoms. I was happy to be working the way I was, making perhaps small discoveries about the structure of molybdenite and other minerals and inorganic compounds. I was eager to understand chemistry better than it was understood in that early period. But I didn't have the idea, I think, that I was going to be the person that clarified this whole question about the chemical bond.
And last, collaboration with other scientists. By collaborating with researchers from different disciplines, Pauling could share his insight and gain valuable feedback. This collaborative approach helped disseminate his discoveries and accelerate progress in the related field.
Okay, so for conclusion, Linus Pauling is one of the greatest biochemists that we know to be exist. His research on the genetic materials in the early 19th centuries helped us humankind to advancing our technologies and understand more about the genetic studies based on his findings in the early 19th centuries. Even though throughout his career he got failure about his research, which nowadays we know to be the triple helix DNA structure. He even published a lot of journals and books to help us understand more about his research and findings. One of the books that he published that I like the most is titled Nucleic Acid.